Welcome everybody. I'm Alberto Vargas. I'm the Associate Director of the program, Latin American, Caribbean, and Iberian Studies program. And it's my pleasure to introduce our speaker today, Janine Richards, who's a PhD student in the Nelson Institute for Environmental Studies in the Environment and Resources program. Janine also has a master's in environmental education from Miami University in Ohio, and she was also recipient of the Tinkerney Summer Research Fund, part of the research. So join me to welcome Janine. She's going to tell us about her result of her research and probably preparation for more research. Okay. Very good. Okay.
Thank you. As Alberto said, I'm in my second year in the Nelson Institute for Environmental Studies, and I was able to conduct my first season of field work last summer thanks to the NAVE Summer Research Grant from LACIS. So I'm going to tell you a little bit about my work so far in coffee farms in Nicaragua, looking at epiphytic plant biodiversity. If you don't know what an epiphyte is, don't worry, we'll get there. So why coffee farms, first of all? I chose to work in coffee farms because I think that they're an excellent model system for an agricultural system that can support a great deal of biodiversity as well, particularly under certain management practices. So coffee is one of the world's top agricultural exports by value. I have seen the statistic that it is the second most valuable globally traded commodity after oil, although there's been some debate about whether that's actually true. But it's a very valuable commodity. It supports the livelihoods of 25 million smallholder farmers worldwide. And coffee is mainly grown in regions that also contain very high levels of biodiversity. And as I mentioned already, there are a number of different production methods that are available for producing coffee that change the impact that it has on that biodiversity. Here's a global map of where coffee is grown around the world. The darker green colors mean higher volume of production. So you can see really high in Central America into Colombia, Brazil, Southeast Asia, Indonesia, and then Kenya, Ethiopia, Central Africa are the major regions of coffee production. I'm going to overlay this with a map of the global biodiversity hotspots. And you can see that those areas are really closely aligned in their area. So these are regions that have a lot of unique species, and for global biodiversity goals, it's really important to be protecting them. In addition, there's also a lot of pressure from human populations in those areas to use the land for livelihood needs. So this is, there are hundreds of ways of producing coffee, but to break it down a little bit from that, here are 5 categories for different types of shading on farms that can be used, going from full sun to what's called rustic coffee farming, which is basically leaving the overstory of the forest and just clearing out in the understory and planting coffee shrubs. Traditional polyculture is a little bit more planted than that. The farmers traditionally will plant other crop plants, such as bananas and citrus, other fruit trees, or timber trees that they plan to harvest later on. So it's very much cultivated beyond the coffee for plants that are useful to the farmers. And commercial polyculture is usually on a little bit larger scale. These two are primarily smallholder farmers. Commercial polyculture is planted overstory. But again, it's a higher diversity of trees. Often they're trees that have other uses as well. Shaded monoculture is probably the most common shade practice on a large scale, and it's usually just 1 or 2 species of trees. Often they're legumes that put nitrogen into the soil to support the coffee plants. And then the unshaded monoculture. To give you a little bit more idea of what this looks like, here's a picture of sun coffee in Costa Rica. And you know, it's kind of like corn around here. It's just a continuous monoculture of one crop. This is one of my research sites. This is planted shade, kind of lower diversity of trees. You can see a lot of bananas in here. There's also some legume trees. But this is a little bit higher diversity than some of the monoculture tree plantings. So I would actually consider this to be a commercial polyculture as well. And this is the other farm where I work, which has a higher diversity of shade trees. Many of them are native forest species that they've either planted into the coffee or were there originally. So I'm looking at epiphytes in coffee farms. Epiphytes are air plants. They grow on the surface of other plants instead of rooted in the ground. And if you haven't spent much time in the humid tropics, then you probably know them best as houseplants. They are orchids, bromeliads, peperomias, ferns. Many of the plants that we grow in pots in our homes are these beautiful flowering plants that you find up in the trees in the places where I'm working. They're ecologically important as well. They're considered keystone members of the ecosystem in tropical cloud forests. They provide nesting sites and material for birds, also food, foraging substrate. They're habitat and food for insects, including some pollinators that are useful to the farmers if they're producing other crops that need to be pollinated. Some of the bromeliads they have what's called a tank structure where they hold some of the water that falls when it rains, and that water can be breeding habitat for insects and also for amphibians. And over time, as the epiphyte communities grow on branches, they'll actually start to form soil up in the treetops that's home for insects, and also there's been evidence that trees are sometimes even putting out adventitious roots into that soil to gather additional nutrients. And so they're important in nutrient cycling as well. So very valuable, but also understudied members of the cloud forest community. My research sites are in north central Nicaragua, near the cities of Matagalpa and Jinotega, is one of the major coffee producing regions of the country. And the farms that I work at are not necessarily representative of the majority of the farms that are found in that region. They are 2 larger farms. They are owned by single families, but they're worked by many people. But I chose them because they are internationally recognized for their commitment to biodiversity on their farms. And also because the farms have, in addition to the coffee farm, they have a forest preserve that is also managed by the owners of the farm. So I would be able to compare the forest to the coffee farm, which was really important for my research. I just wanted to tell you a little bit about the importance of coffee in Nicaragua, both economically and culturally. It's been the top agricultural export since 1870, and it's currently the third export in value overall. It's supporting some 45,000 families and 95% of those are on small farms. There's actually a much higher proportion of smallholder farms in Nicaragua than in some of the surrounding countries. And some of that is due to land redistribution that happened following the revolution that took land away from large landholders that had been affiliated with the Somoza regime and gave it to the people that had been working the land previously. And there is also a long history of cooperatives, which really support the economic infrastructure necessary for smallholder farmers to be able to sell their coffee profitably. And the first cooperative was actually formed by Augusto Sandino in the 1920s. And coffee is culturally important as well. I just picked out a picture from during the revolution in the 80s. Some Sandinista revolutionaries, also students and even international volunteers came to help get in the coffee crop in Nicaragua during the revolution so that it wouldn't go to waste as the revolution was happening. Deforestation in Nicaragua, like in much of the world, is a major loss or a major threat to organisms that depend on forest. So 1983 to 2011, you can see a large loss in forest cover across the country. The red circle is the area where I'm working in the coffee growing regions. So you can see that there's still a fair amount of forest cover there, but a lot of it is fragmented into very small pieces of forest that are disconnected with the rest, with other patches of forest. So for organisms like mammals or amphibians or plants that don't move around very easily, it's hard for them to get from one patch of forest to another. And that's actually one of the great possibilities for shade coffee in biodiversity conservation is the ability to connect fragmented pieces of forest with a more continuous canopy of trees so that some of these animals and even plants can migrate more easily among the forest patches that are left. This is just to give you a kind of a visual of what that landscape looks like. This is a vista from one of the farms that I work at, and you can see that there's quite a bit of forest cover, but there's also a lot of open patches of land as well in between. Here are the locations of my two farms. They're fairly close together in the scheme of the country. And you can see from the pictures that they look pretty, like they have quite a few trees, pretty verdant. And that is true when you look at the aerial photo as well. I don't know how well you can see this with the lights in here, but there's definitely some darker concentrations of darker green representing forest in those two areas. And those are largely the forest preserves at those farms. The research questions I was addressing last summer when I was there were basically looking at the comparison between epiphyte populations in the forest and epiphyte populations in the coffee farms. I was also gathering data on particular tree and site characteristics that might influence those populations and what species are parts of those communities. And then I also gathered data on life cycle, where a plant was, whether it was an infant, juvenile, of reproductive age, to kind of understand whether epiphytes are able to complete their life cycles within the coffee farm, not just established there initially. And I'm not really going to talk about that because I have not analyzed those data yet, but I will talk about the first two questions. I was climbing trees using single and double rope techniques to gain access to the canopy, and which was pretty fun. I was installing the ropes in the trees with a giant slingshot, and then climbing up on ropes, and well, I'll go to the next picture. And then I set up 3 vertical plots along the trunk of the tree where I recorded all of the epiphytes that were present. And I also recorded the diameter of the segment, how much, I estimated how much moss and lichen were covering the bark in that segment, and also recorded the number of branches that were diverging from the trunk. For the trees, I recorded how far they were from the edge of the forest, some size estimates of the tree, including the diameter, the height, and the crown width and length. I recorded the density of the light coming through the canopy at the base of the tree, some qualitative measures of bark texture, and then I also used temperature and humidity data loggers in the farms and in the forest to record temperature and humidity over time during the time that I was there. For the epiphytes themselves, this is one of my 10 to 11 meter plots in one of my trees. I recorded what species the plant was, how many of them there were, the age class, which I talked about before, whether there was evidence of reproduction, either flowers or fruit, and what substrate they were actually growing in. I sampled 42 trees total with 126 vertical plots in them. And at each of the farms, I did some trees that were in the coffee fields and some trees that were in the forest. And that's, as I go into my results, that's kind of how I'm going to break it up, is farm, forests at each of the farms. So my results so far, and again, these are pretty preliminary, are that the strongest predictors of species richness and abundance are similar, but there are a few distinctions. For species richness, so the number of species present in one of those plot segments depended the most on the percent moss cover, where it was located, the farm, and then whether it was in forest or coffee. How many branches were coming out of the trunk in that segment, and how far the segment was off the ground. For the predictors of just how many total epiphytes there were in those segments, moss cover again, the size of the tree based on its diameter, the number of branch junctures, the height off the ground, and the location. I looked at light levels, and this is based on the amount of light that was hitting the ground at the base of the tree. And what I found is, so blue and pink are the coffee plots at the 2 farms, and then green and yellow are the forest plots. But across the board at each site, I found a decline as light increased. So as the amount of light hitting the ground at the base of the tree increased, the amount of the species richness of epiphytes in that tree decreased. I also found the highest and the lowest levels of species richness in the 2 forest plots. So here is my highest level of species richness is at the El Jaguar forest plot, and my lowest level of species richness is in of diversity is in the Selva Negra forest. And this is pretty counterintuitive. I would have expected a pattern like these top 2 lines are the El Jaguar forest and the El Jaguar coffee. And I would have expected something like that, where the forest had a little bit more species richness than I saw in the coffee. But at Selva Negra, I saw the reverse pattern, where there was actually many fewer species in the plots that I had set up in the forest. But in analyzing the data, I realized that this is probably an error in my sampling method, because the trees, this graph is tree height. So the trees in the Selva Negra forest were considerably taller than all of the other trees that I sampled. and I was only sampling the bottom 10 meters of the tree. So I wasn't getting all the way up the trunk in those trees to, you know, the places where the branches were starting to diverge, where the highest species richness was found in the other trees. So I think I was just missing where that diversity was in the tree. And I regressed tree height and species richness and found that yes, there is a pattern of declining species richness with tree height. But again, I think that's an artifact of the sampling method rather than an actual pattern. So thus far, my conclusions are that there is no pattern in epiphyte abundance and richness between the forest and coffee. However, that may be a sampling artifact. And when I just look at the El Jaguar site, where the trees are of roughly equal heights in both in both the coffee and the forest, there is a pattern of greater species richness in the coffee. And even if this is true, that you can't tell apart the forest and the coffee, that's actually a good result for conservation. I mean, that's what we want to see, is that you're getting just as high or almost as high levels of biodiversity in the coffee as in the forest. And then at the site level, I found that species abundance and species richness were related to light levels, the height off of the ground, the size of the tree, and the number of branches that were coming out. So my future work, hopefully this summer and beyond, will be to revise the sampling methods to reflect height differences and go back to some of the trees again, or maybe find some new trees in the Selva Negra forest site. I also am hoping to expand my sampling beyond the trunk to look at epiphytes that are growing on the inner and outer branches, so that I can get a fuller picture of the entire population that's within the tree. I will be also measuring traits of individual plants, such as leaf thickness, and, well, I don't have a list yet, but some traits that are characteristic of drought tolerance or sun tolerance that I would expect to find in higher proportions in the coffee farms than in the forests. And in kind of the last stage of my research will be broadening beyond these two farms and looking at a number of smaller farms as well, to get kind of a spectrum of how biodiversity varies across the types of management practices that are most commonly seen in the farms in that area of Nicaragua. So I want to say thank you again to LACIS for the NAVE Summer Research Grant that allowed me to begin this research. I also received funding from the CIAS mini grant program. I'll be using funding this summer from the Botany Department Demeter Award, and last year I was funded by a Potter Distinguished Graduate Fellowship. Thank you. And this is a mural of the Selva Negra farm that's painted on one of their buildings that I think is a beautiful model of agriculture and biodiversity together. Yes?
2 questions. Is there a Department of Natural Resources in Nicaragua that's, are you doing this alone, or is there a government agency that's also interested, and also is there a university that has a focus in this area and and students are working on this? Or
I have not found the students working on it, and I'm not working with a department. I have been working with the herbarium there to identify the plants that I'm finding.
Okay.
And I'm hoping at some point that I may be able to hire some local field assistants and tap into some students that way.
Is there any interest in working with universities, you know, trying to get them interested in this, or you don't?
I would love to. I haven't found the right connections yet.
Just kidding.
Yeah.
Could you tell us a little bit more about your sampling technique? So how do you select individual trees to
Sure.
study? And then the second question is, so part of your findings, moss was an important predictor of richness. So what predicts moss?
I think it's related to humidity. So the drier sites or sections of the trunk that were in direct sun exposure have less moss. And I haven't run numbers on that yet, but that's my sense just from observation. And from what I know of moss, that makes sense. And then for how I selected the trees, I randomized my selection based on random numbers generation. It was actually one of the harder parts of my research, where I had a list of random numbers, and I was using them to decide which direction to walk in and walk a certain number of paces, and use whatever tree was closest to where I stopped.
So you use GPS to know where is the tree that you were sampling?
Yeah. After I found the tree that I was going to sample, I made a GPS point so that ultimately I'll be able to look at all of those points on a map and calculate how far they are from the forest edge, and I can use that as a variable also. Yes.
Why Nicaragua?
That's a good question. I'm really interested in the social history there, and how the revolution played into biodiversity conservation today, and also into agricultural decisions. And I had traveled there previously. One of the Selva Negra farm, this farm, I had visited on previous occasion, and I knew that I would be perfect for what I was thinking about doing. So that was a major factor in choosing Nicaragua.
I have a couple of questions and a comment. So following on this trend of questions that they asked, by randomization, you found the tree that was going to be your sampling site. So that means that there were probably different species, and mostly maybe in the forest, more likely. Do you think that because a different species can grow at different speeds, and the trunk can have different texture, so that there could be confounding factors in, between two comparisons of individuals that might also be throwing your data into some of those?
Sure. Yeah. There are definitely. That's definitely the case. I mean, the tree diversity in the forests here is very high. The research that I've read shows that the bark texture seems to be more important to epiphyte populations than the tree species. So I was recording the bark texture to try to see if I can see how that is in the analysis, whether it's a factor or seems to be a factor or not.
Because I think that having too many variables, but I would be concerned that if you want to expand to smaller farms later, you increase also other variables in sizes and management, plus all these variables on the shade, the texture, the age, the size, the diameter of the trunks, the branching, that it can be very hard to then conclude something that maybe you can think about design where you do some very, like, if you can choose, if you know that there are similar species, at least in the forest in the two locations, to reduce some of the variables, and so you can have some comparisons like that. And I was wondering if there could be a possibility of sampling some diversity in the ground, because all the trees have a ground, like the base, so you eliminate if there's branching, if there's higher or if there's, texture. And you are measuring the light intensity at the bottom anyway.
Welcome everybody. I'm Alberto Vargas. I'm the Associate Director of the program, Latin American, Caribbean, and Iberian Studies program. And it's my pleasure to introduce our speaker today, Janine Richards, who's a PhD student in the Nelson Institute for Environmental Studies in the Environment and Resources program. Janine also has a master's in environmental education from Miami University in Ohio, and she was also recipient of the Tinkerney Summer Research Fund, part of the research. So join me to welcome Janine. That she's going to tell us about her results of her research and probably preparation for more research. Okay. Very good. Okay.
Thank you. As Alberto said, I'm in my second year in the Nelson Institute for Environmental Studies, and I was able to conduct my first season of field work last summer thanks to the NAVE Summer Research Grant from LACIS. So I'm going to tell you a little bit about my work so far in coffee farms in Nicaragua, looking at epiphytic plant biodiversity. If you don't know what an epiphyte is, don't worry, we'll get there. So why coffee farms, first of all? I chose to work in coffee farms because I think that they're an excellent model system for an agricultural system that can support a great deal of biodiversity as well, particularly under certain management practices. So coffee is one of the world's top agricultural exports by value. I have seen the statistic that it is the second most valuable globally traded commodity after oil, although there's been some debate about whether that's actually true. But it's a very valuable commodity. It supports the livelihoods of 25 million smallholder farmers worldwide. And coffee is mainly grown in regions that also contain very high levels of biodiversity. And as I mentioned already, there are a number of different production methods that are available for producing coffee that change the impact that it has on that biodiversity. Here's a global map of where coffee is grown around the world. The darker green colors mean higher volume of production. So you can see really high in Central America into Colombia, Brazil, Southeast Asia, Indonesia, and then Kenya, Ethiopia, Central Africa are the major regions of coffee production. I'm going to overlay this with a map of the global biodiversity hotspots. And you can see that those areas are really closely aligned in their area. So these are regions that have a lot of unique species, and for global biodiversity